Welcome back, everybody. I hope you had a very good brief break. And thank you for sticking around for the third of today's sessions, which today we're going to be looking at genomics, snails, immunology, and also some mapping. So first up, I'd like to introduce Dr. Jonathan Shortt, who's going to be talking about population genomic analyses of schistosome parasites, and he's going to highlight critical challenges facing the endgame elimination efforts. So over to you, Nick, Jonathan, sorry.
Hello, everyone. My name is Jonathan Shortt. I'm a research instructor at the University of Colorado Anschutz Medical Campus, and I'd like to thank the meeting organizers for the opportunity to share with you some of the research I completed during my graduate work on what we can learn about schistosomiasis control efforts using population genetics. The work I'm presenting today was recently published in Scientific Reports, and the QR code on this slide and on the final slide should take anyone interested in learning more directly to the manuscript. I actually did my graduate work in human medical genetics with a bunch of people who know very little about schistosomiasis, so I'm used to beginning these talks with an introduction to the devastation caused by schistosomiasis throughout much of the world. Given the nature of this meeting and your expertise on the subject, I'm happy to skip over some of those details, but do wish to start out by highlighting what you probably already know, that billions of people throughout the world live in areas where schistosomiasis is endemic and live at risk of infection. Even though the human race has lived with schistosomiasis for thousands of years, I'm encouraged by the progress that research such as that presented here today has brought about. One example of such progress can be found in China, where schistosomiasis infection prevalence has dropped 99% since the 1950s due to their integrated control program, which includes education, snail control, and importantly, chemotherapy. Despite this progress, schistosomiasis transmission hotspots remain in regions of China. In a recent survey that our collaborators conducted in 36 villages in Sichuan province from 2007 to 2010, nearly one-third of villages had infection prevalence higher than 10%, 7 of which had infection prevalence higher than 10% each year of the survey, despite intervening treatments. The figure on this slide predates that survey, but further illustrates that schistosomiasis is endemic or emerging in many regions of Sichuan province, and the reasons for these persistent infections are not well understood. We're taking a population genetics approach to understand how schistosome populations persist, and the first question we'd like to know is what genetic population structure, if any, exists across the region, because that will help identify potential sources of infections in remaining pockets of transmission. For example, if samples collected from different villages appear closely related, this suggests that these villages share a common source of infection. But if samples from different villages appear different from one another, this suggests multiple unique infection sources. An understanding of population structure will also enable us to view the spread of schistosome infections throughout the region to better assess regions or even individuals that may benefit from increased treatment focus. And these same questions can also be asked at a village level or even a host level as opposed to the region level. These types of questions are not necessarily straightforward to answer, so I'd just like to highlight a few key points that enable this type of research. First, we're interested in the population structure of infective parasites that are living within human hosts, so there isn't a practical way to sample them at population scales. However, mating pairs from Schistosoma japonicum, which is the primary infective schistosome species to humans in China, can shed up to thousands of eggs each day, and these are more readily available to sample. For our research, Myricidae are collected on site after hatching, and individual Myricidae are dropped onto these FTA indicator cards with a small amount of water where the DNA from the Myricidium can be preserved for years under the right conditions. 2. There's limited DNA within a single Myricidium and not enough to prepare sequencing libraries. To overcome this problem, we're using whole genome amplification to generate micrograms of DNA from a single Myricidium. Fortunately, whole genome amplification is relatively inexpensive, and it's fast enough to do in the lab so that it doesn't become a bottleneck. 3. High-resolution population structure requires many genetic markers. Many genetic analyses in non-model organisms in the past have relied on amplification of polymorphic microsatellites, but these assays can be expensive to develop and don't necessarily scale well to the number of markers that we require. We use double digest restriction site associated DNA sequencing, which uses restriction sites to select and amplify thousands of the same loci in each Myricidium for sequencing. In the interest of time, I won't go into more detail on ddRADseq, but it's relatively inexpensive and scales well, and in our hands we can go from a Myricidium on an FTA card to sequence data in about a week's time, so we've been quite happy with it. We applied whole genome amplification and dd-rad-seq to Myricedia collected from 12 villages in 2007, 2008, 2010, or the summer or fall of 2016. Villages range in distance from under 1 kilometer to just over 25 kilometers. After quality control, we ended up with 200 Myricedia collected from 34 hosts. Our sampling scheme was to sequence at least some Myricedia from numerous villages with multiple hosts at multiple time points in at least some villages. This scheme is enough to get a good sense of schistosome population structure in the region, and can also generate at least anecdotal evidence for how the methodology might be applied in a more exhaustive manner to ask questions about the frequency of retained infections, and if the number of infective mating pairs within a host can be estimated from sampling their progeny. The data I'm presenting today is based on nearly 34,000 single nucleotide polymorphisms sequenced on dd-rad-seq loci. Principal components analysis finds the axes of greatest variation, called principal components, from a multidimensional space, and when used with genetic variation, it separates samples by how different they are from each other. When applied to our Myricedia, villages separate from each other across the first 5 principal components, with the first 2 principal components shown here separating the 2 most distant villages, indicated by the color of each point. This result indicates that there is regional population structure, that infections from one village can be distinguished from infections in another village. This regional structure also means that we can identify likely regional sources for new infections. We also built a neighbor joining tree using allele sharing distance between each pair of Myricedia. In the tree on the left, leaves are Myricedia colored by the village of collection, and you can see that villages tend to cluster together. This is an indication that variation between the villages is greater than the variation within the villages. The same neighbor joining tree is shown on the right, but colored by time point, and shows that structure doesn't appear to change much over collection time points, even though schistosomiasis positive individuals were referred for treatment after each collection. There are other interesting questions to ask about region-wide population structure, but we ultimately want to understand the source of infections within each village and within each host if possible, and that requires that we somehow use this data to get at family structures and pedigrees. To do this, we took a pretty straightforward approach and asked what proportion of alleles is shared between all pairs of Myricedia. Since rare alleles are more unlikely to be shared by unrelated individuals, we used only alleles with minor allele frequency less than 0.1 for this analysis. This figure shows distributions of rare allele sharing between pairs of Myricedia collected from the same host in green, from the same village but different hosts in gray, or from different villages in blue. We find that samples within a village are generally more related to each other than samples from different villages, and frequently share alleles at a level that might be expected for cousins. Samples within the same host are also frequently sharing at a cousin level, but also show increased relatedness that is consistent with first or second degree relatedness. To get a better sense of relatedness, we calculated the posterior probability of different degrees of relatedness between each pair of Myricedia, using the mean proportion of allele sharing between highly related clusters of Myricedia collected from the same host and at the same time point to create a model for the proportion of alleles shared between siblings. We calculated the posterior probabilities for each degree of relatedness from first to fourth degree between each pair of Myricedia. Here, I'd just like to point out that among the different types of first-degree relationships, namely sibling relationships and paired offspring relationships, we can only have siblings in our data since the parents live within hosts and cannot be sampled. Distinguishing between different types of second-degree relationships, which could include half-siblings, double first cousins, as well as avuncular relationships in our data, is not as straightforward and creates opportunities for interesting work in the future. In any case, the posterior probabilities do allow us to make some interesting observations in this dataset. First, we observe multiple hosts infected with multiple mating pairs, observed by independent clutches of siblings within the hosts. This slide shows 2 such hosts, with Myricedia collected from a single host arranged as a circle, and bands connecting different Myricedia are colored according to the posterior probability of first degree or sibling relatedness. Groups of Myricedia that are connected by bands form sibling clutches, and multiple clutches within a host can give a minimum estimate of the number of infective pairs within a single host. We also find some evidence for infections that are retained over time, which show up in the data as sibling Myricedia that are collected from different time points. I'll note here that people who tested positive for infections were referred for treatment, so we didn't necessarily expect to see retained infections, though it is possible that some infections were retained due to noncompliance. Noncompliance should not be the case for 2016 infections shown on the left, when directly observed treatment had been implemented. The degree to which these retained infections are the result of noncompliance with treatment suboptimal dosing, drug resistance, or something else warrants more research. Here we show posterior probabilities for sibling relatedness and third degree or cousin level relatedness for the same Myristidia collected from 3 separate villages. As seen in previous slides, sibling clutches are frequent within individual hosts in the figure on the left. On the right, we see that third degree relationships dominate between hosts within the same village comparisons. This high level of allele sharing within villages indicates that parasite mate choice is often limited to relatives during the reinfection process, and implies that following treatment, infections in a village may have been reestablished by a small number of genetically unique schistosomes, likely reflecting the effectiveness of local schistosomiasis control programs. These results also suggest that long-term elimination may require identification and targeted treatment of remaining local parasite sources. So I'm coming at you live now because the narration on my conclusion slide didn't come through. There are just a few quick conclusions from this work. First, schistosome populations in Sichuan, China appear highly structured, with populations from different villages appearing fairly distinct from other populations, even if they are not separated by a large distance. Second, infections within villages appear to be driven by local transmission and reinfection, since treatment cycles between collection time points do not greatly alter population structure. Third, local parasite reservoirs are not fully eliminated, with some human hosts retaining infections even after referral for treatment. This leads to limited reinfection sources within villages and short transmission routes between human infections. Because of retained infections, it's possible that human hosts participate in maintaining infection reservoirs, though we do not exclude participation from other non-human mammals. Finally, to really eliminate infections from villages, we need to focus on confirming that infections are eliminated after treatments and ensure that other sources are also eliminated. And can we go to the next slide, please? As with any work, I owe thanks to many collaborators. This is really the cross-collaboration between 3 different institutions. My university, CU Anschutz, University of Texas, Arlington, and collaborators in China as well. Thank you for your attention.
Brilliant. Thank you very much indeed, Jonathan. And sorry, your image suddenly swapped with Nick's video when I was talking to you, sort of. That's why I suddenly said, I was like, that's not right person. the trials of Zoom, I suppose. Anyway, we've got a couple of questions I know from the panelists, actually. So Nick, having introduced you briefly by accident earlier anyway, I know you have a question. Do you want to just turn your mic on and ask it?
Yes, I can do that. So it's really cool, Jonathan. I'm curious if you know anything about the travel and migratory patterns between the villages that could be mapped onto the population structure and explain some of that.
So I don't have that data for these villages particularly. There aren't like necessarily roads connecting each of them, and there aren't really bodies of water or streams between them. But again, we see that like it doesn't look like there are many cross village infections. It looks like kind of each village has its own flavor of schistosomes, with closer villages obviously having some of the same, a little bit similar flavors. But that hasn't been mapped into this analysis, but it's definitely something that I think we're going to try to do in the future.
Cool, thanks. And Bonnie, you also had a question. Do you want to ask that?
Yeah, hi Jonathan, thanks. So various people on this call today has been working on population genetics of parasites, and we've always had this question about, do you sample more hosts or do you sample more Myristidia within a host? So one of the questions I have is, do you think a pooling strategy on analysing lots of Myristidia from a single host could be done within this kind of analysis?
I think that's definitely possible. It would kind of limit some of your resolution because you wouldn't know which loci necessarily come from which Myristidia that were pooled into the same pool. Although I guess if you were to look at individual haplotypes, you could count how many haplotypes are at any given locus in the genome, and you could get, I guess, a minimum estimate for the number of kind of distinct haplotypes are in there and make some sort of guess about the total number of mating pairs or total number of unique organisms that you've actually sequenced. Could be interesting. it would definitely, like, to look at between village population structure would definitely work, and you could definitely sequence more.
Great, thank you. Quick question from the Q&A. It's a comment really, but I think you'd like your view on it. So it's from Julio De Leo. The small number of genetically unique schistosomes makes me think to a founder effect. So of a few or a single infected individual or individuals seeding schistosome parasites in the village, and wondering what you thought about that.
Yeah, I mean, I think that's really consistent with the data that we've collected. And because they're so, I mean, a lot of the schistosomes we've sequenced within a village appear to be kind of related at kind of like a cousin level. And that really kind of indicates that, you know, so if you think about your cousins, you share the same grandparents. So that's a very short transmission route between human infections. And we didn't sequence any of the animals in these villages. So we do think there's very limited reinfection. And that also kind of highlights that the treatment is mostly working. So when they go in and test these villages, they give everyone praziquantel, and then when they leave, they come back a few years later and retest for infections. And it does show that I think that praziquantel is mostly working. It's just a few infections are escaping and then reinfecting the whole village is kind of what we think is happening.
And I actually had a quick question about that as well, if there are no others in the Q&A. With the really strong structure by village, I mean, obviously, it depends how dispersed they are, but what kind of that might say about the zoonotic hosts and the movement of those. And I know in different regions, different zoonotic hosts are important, but do you think with that level of structure, it sort of helps indicate which hosts are sort of driving reinfection in the humans?
Yeah, I mean, I think that would, you know, if wild animals crossing between villages, it wouldn't seem that they're contributing much, at least in this scenario, at least in this region. It does seem like they're more kind of the human adjacent animals, maybe domesticated cows or dogs or rabbits, you know, something that's in the village and living with the humans is kind of what I would think. But, you know, there's interesting questions to ask. This is just really just kind of you know, we sequenced a lot and we were able to gather some anecdotal evidence for a lot of interesting questions to ask in the future. So,
well, we hope you come back and ask them, because we will forgive you for moving on to human research at the moment, but we hope that your heart will remain in schistosomes. So thank you.
Thank you.
Brilliant. Well, next up, we have Dr. Roseli Tuan, who is a senior scientific researcher and working in São Paulo. And she's going to be talking today about modeling the present and future distribution of Biomphalaria species along the watershed in, now if I can say this correct, the Middle Paranapanema region in São Paulo in Brazil. So thank you very much. Over to you.
Hello, I am Roseli Tuan from São Paulo State, Brazil. I'd like to express my appreciation to GSA for the invitation, giving me the opportunity to share with you my experience in malacology. I want to also to stress that Raquel Gardini Sanchez Palacio, as my PhD student, and the partnership with Universidade de São Paulo made possible the development of this work, which resulted in a present and future mapping of 5 Biomphalaria species, which inhabits a special region in São Paulo called Middle Paranapanema region. What grabbed my attention to this region here, the Middle Paranapanema River region, was the presence of the three mainly hosts for Schistosoma mansoni in neotropical area, Biomphalaria glabrata, Biomphalaria tenangophila, and Biomphalaria stramina, and also Biomphalaria peregrina and Biomphalaria occidentalis. Schistosomiasis is not so important today as it was in the past. Some changes happened, and today we have few cases of people locally infected by Schistosoma mansoni. So it's not a region which has a high importance for the disease. but it has a high importance in terms of malacology and in terms of the possibility of expansion of Biomphalaria glabrata, considered the most susceptible species to Schistosoma mansoni. To develop this work, we sampled snails in 114 freshwater bodies inside 4 municipalities: Ourinhos, Ipaúçu, Ribeirão do Sul, Chavantes, and Assis. These municipalities are considered um, risk areas for the expansion of this species, mainly Biomphalaria glabrata. We sampled more than 600 sites and used GPS coordinates obtained in all the more than 600 sites where we collected the snails. It gave to us a chance to produce digitalized maps. Well, to we used Maxent as a source, as a program for modeling species distribution. And Maxent needs presence only species records. And for us, it means identification. And we performed the identification of the snails throughout morphology and DNA barcoding. And we can see here the results we obtained in the sampled areas. And what we see here is that both morphology and DNA barcoding both have the same results. Both resulted in a high confident result for species identification. Here is the map produced with the data set of snails and GPS coordinates. And what we can see here is there is a high probability to sample Biomphalaria glabrata just in Ourinhos, in specific sites that which we considered hotspots for the suitable sites, hotspots for suitable sites for the Biomphalaria glabrata species, and it's a distribution that is different from the past, because in the past the chances are that you could find Biomphalaria glabrata spread over the area. Today we didn't see this. And it's very different from Biomphalaria occidentalis and Biomphalaria peregrina species that spread over the area. It's interesting also to notice the distribution of Biomphalaria tenagophila. In the last years, in the surveys in the region, we could find Biomphalaria glabrata in many other sites. Today, we found Biomphalaria tenagophila just in Ipaúçu, in a more frequent in Ipaúçu, and it's a thing that had changed a lot. The region was recognized at not only a site for Biomphalaria glabrata, but also a site for Biomphalaria tenagophila. The taxonomical composition of the region is complex. We need to understand better how the species, the Biomphalaria species, and the other species of planorbid species relate, compete each other, because sometimes, and most of the times, we can find them in the same habitat. I would not be able to explain in details the methodological and statistical approaches we use it here, but I can explain to you, I want to show to you the logical steps we use it to have the maps, to have the actual and the future maps for Biomphalaria distribution. First of all, we grabbed bioclimatic variables, hydrological variables, soil types, and altitude in database in the internet. And this bunch of variables were analyzed to reduce to more statistically significant variables in PCA, which is a principal component analysis and statistical analysis, and used in Maxent this data under two different scenarios in terms of greenhouse gas emissions. A 2.6 scenario where we have a strongly decline of emissions during the years, and a scenario of RCP of 8.5, which characterize a drastic scenario, which the rise of temperature to more than 4 degrees, and it's a strong scenario. So what we have here, it's the 8 mainly variables selected through principal component analyze, consider the variables more significant that have more impact on species distribution. What we see here is for Biomphalaria tenagophila and Biomphalaria occidentalis, precipitation, rainfall is the main component of the climate that can interfere with the distribution of both species. It's different from Biomphalaria glabrata, stramina, and peregrina, where the PCA analysis showed that temperature are the most important climate factor that might impact the distribution of these species. These are the results, the maps that resulted from the analysis in Maxent. Here, the present distribution of the 5 species. Here, the distribution in a scenario of low increase in greenhouse gas emission, and in the drastic scenario with a high increase in GGE, with consequently a rise of temperature. First, what we see here at first, it is that that different species have different patterns of distribution, not only today, but over the years, under the impact of climate change. Biomphalaria tenagophila, Biomphalaria occidentalis, and Biomphalaria peregrina seems to maintain the distribution that we find today. We cannot see an expressive expansion or reduction in the distribution of these species under the rise of temperature, for example. And especially for Biomphalaria occidentalis and Biomphalaria tenagophila, genetics might be an important explanation. As we have observed in many occasions, Biomphalaria tenagophila and Biomphalaria occidentalis are so much depauperated in terms of genetic diversity. And we know that adaptation is linked to genetic variability. So if in terms of genetic variability, I should say that Biomphalaria tenagophila and Biomphalaria occidentalis should not be able to expand, to adapt to climate change in a way that they expand the range of their distribution. They tend to maintain what we see today. And this kind of genetic poverty should drive the colonization of the species over the area in the next years. It's different from Biomphalaria glabrata. When we see Biomphalaria glabrata, and we, in the next slide, we can see only Biomphalaria glabrata distribution in now and in the future. We notice that there's a probability of expansion of the hotspots, of the small hotspots over the area. But what's more important, that the chance is that the species cross the limits of distribution that we see today to the north and east and west of Paranapanema region. What might explain this? Well, the resilience of the species, the adaptation to different temperatures might be one of the most important factors that help the species to survive under climate change. But another kind of explanation is based on the soil composition, because the Paranapanema region has two different soils in their composition, and Biomphalaria tenagophila is today in sites where rich clay soils exist. In the north of Paranapanema, we have more sandy components in the soil, and in the future, it seems to me that Biomphalaria glabrata might have the ability to survive in these sandy soils more than today. And in terms of greenhouse gas emissions, in Brazil today, agriculture is the most important factor which contribute to the increase of greenhouse gas emissions. When you look to this table and we see the increase, the 3 decades, the impact of 3 decades of land use and land cover and impact on land cover, we see that agriculture and the use of land to livestock changed a lot in the last 3 decades. Also, the urban infrastructure. And all these factors should improve the colonization areas, should improve the possibility of the species to cross the boundaries of where they colonize today to new regions. For example, to Amazon region. For example, Biomphalaria glabrata today can be found in Amazon region. So when we know that the Amazon is being deforested, so it's important for Schistosomiasis to analyze the possibility of the expansion of Schistosoma mansoni to this area through the expansion of Biomphalaria species. Thank you.
Obrigada. Thank you very much indeed for that presentation. I don't believe we have any questions in the chat, so please do put your questions in the chat, and she can answer them as time goes by. But in the interest of time, I think we might just move on to the next talk. Just to introduce Dr. Derick Osakunor, who did his PhD at the University of Edinburgh and is now at the Children's Research Institute in Washington, DC. And Derick will be talking about some of the work he's done on immunology, particularly in primary school children, and looking at schistosoma haematobium and the effects in energy and purine-related metabolism. So thank you very much, and over to you, Derick.
Hello everyone, and thank you so much for giving me the opportunity to share with you my findings from a study on host metabolic alterations from early schistosoma haematobium infection in preschool-aged children. So for schistosomiasis, why the interest in preschool-aged children in the first place? So schistosomiasis is a vicious cycle with devastating consequences, including malnutrition, poor cognition and physical activity, most of which occur early in life from exposure, and this can go on right into adulthood with sometimes irreversible chronic morbidity, as you can see on the slide shown here. So up until 2010, where the WHO issued revised guidelines for the inclusion of preschool-age children in treatment campaigns, most of these kids hitherto were excluded from treatment programs in most schistosome endemic areas. However, when you go on the field, what you see is like the image I have on the slide, where people across all ages are exposed to infected water and can get schistosomiasis. Some of the reasons attributed to the exclusion include the lack of a child-friendly formulation for praziquantel, a lack of a consensus on dosing and safety of the current tablets, a lack of a coherent strategy to access and treat as with school-age children, and a lack of understanding of risks, infection, disease, and treatment dynamics, and finally, operational challenges with diagnostics. Now, some of the consequences of this exclusion includes the fact that these kids become reservoirs for transmission, they have longer lasting infections with heavier burdens, there's severe morbidity and sometimes mortality, and all of this put together results in serious inequalities in healthcare, in addition to a low research focus on preschool-age children when it comes to schistosomiasis. So moving forward, studies have shown us that host-parasite relationships are central to parasite survival and pathology. And so the crux of schistosomiasis control has been to reduce morbidity associated with infection. So what if we are further able to understand this host-parasite relationship and use that to provide further information on what the host's responses to infection and infection burden is, the impact on host health and parasite survival, the effect of current treatments on host responses, as well as the implications on the current and future health, we'll be able to find new ways for control and to reduce disease consequences. So this is where metabolomics can play a key role. With a metabolome being a direct downstream output of the genome and an upstream direct mirror of environmental influences on the body, we can determine what the systemic impact of schistosome infection is on a preschool child if we can determine the change in metabolic profiles at infection and post-treatment. Now, such longitudinal studies are limited in humans and more so in preschool-age children, and even more so when it comes to haematobium studies. And so at the time of conducting this study, this was the first of its kind. And so in rural Zimbabwe, we followed a group of kids who had never had schistosome infection for a period of 3 months, at which we screened them again for infection, and a group of children who were positive for infection received praziquantel treatment and were screened again after 3 months to ensure that they were cured. Now, at all these time points, 0, 3 months, and 6 months, these kids were screened for infection, and blood samples were taken for metabolic profile measurement via mass spectrometry. I'll move on now to show you what we found in the study. We analyzed the baseline metabolite profile data to determine if there were any baseline variations in metabolite levels. Now, this was important to do because in determining what the change in metabolite profiles were with schistosome infection, you can account for any of these baseline variations if any at all existed. A MANOVA model showed that sex but not age influenced metabolite levels, and a fold change analysis showed a greater than 2-fold change increase in metabolite levels in males, with a general pattern of increasing concentration in males as compared to females. So we further adjusted this baseline data for age and used the residuals in a multivariate analysis model to determine which metabolites specifically were accounting for this male to female group separation. The OPLSDA model, as shown on the left-hand side, clearly indicated or showed this separation, and its corresponding S-plot that shows all the metabolites in question, their within and between group variations, and using specific cutoffs, showed creatinine, citrulline, sarcosine, and GABA as the most significant metabolites accounting for this between group separation amongst males and females. At 3 months post-infection, we calculated the change in metabolite profiles between baseline and at infection, and used this data in a MANOVA model to determine the influence of schistosome infection on metabolite profiles, accounting for age, sex, and their interactions. Schistosome infection did indeed significantly influence metabolite profiles at infection, and a fold change analysis showed greater than 2-fold change increases or decreases in metabolite profiles amongst infected individuals, and a pattern correlation analysis confirmed this pattern. So like we did with the baseline data, we adjusted for age and sex and used the residuals in a multivariate analysis model to determine specifically which metabolites were contributing to this infected-uninfected group separation. As you see on your left-hand side, the OPLS-DA model clearly showed this separation, and its corresponding S-plot, which was used in conjunction with specific cutoffs, i.e. the P-core and the VIP values, showed that AMP, 3-phosphoglyceric acid, ADP, and glucose 6-phosphate were the most significant metabolites contributing to this between-group separation. So if indeed these metabolites are increased post-infection, then it is plausible that upon regression analysis, we found that as infection intensity or infection burden increased, the levels of these metabolites also showed an increasing trend. Now, even more interestingly, an analysis of children who tested positive for schistosome infection and were followed up 3 months later showed that these significant metabolite alterations returned to near pre-infection levels following curative treatment with praziquantel. Now, as a follow-up to determine what the exact systemic impacts of the alterations we observed in this study are, we conducted a pathway analysis which showed that the most significantly impacted pathways were involved in energy and purine metabolism in the host. So with the results I've shown, we propose a model as to what exactly is happening in the host system. With regards to energy metabolism, we know that the schistosome worm can use up the host glucose levels up to an equivalent of its dry body weight every 6 hours. This means that infection with the schistosome worm will result in significant decreases in host glucose levels, ATP levels will decrease, and the normal body response will be an increase in ADP and AMP levels. In attempts to compensate for these low levels and generate alternative sources of energy, G6P and 3PG levels will also increase to feed the PPP pathway and the fatty acid synthesis pathway. With regards to purine metabolism, experimental studies also show that the schistosome worm lacks de novo synthesis of purines for its survival and uptake, and so it resorts to its endogenous alkaline phosphatases and other phosphohydrolysis to cleave phosphate molecules like ADP, AMP, and G6P to generate these purines. Now, cleavage of these molecules also results in free adenosine, which has also been shown in experimental studies to be involved in immunomodulation of cells and cell activity. And this is not surprising given that schistosome infection is associated with a significant immunomodulation and a pronounced Th2 type response. So in summary, we've shown that the first haematobium infection in preschool-aged children results in significant host energy and purine metabolic alterations, and these changes correlated with infection intensity and resolved 3 months post-curative treatment with praziquantel. These have implications for reduced host energy as well as host system immunomodulation and parasite survival. Our findings have insights into host pathogen interactions which are consistent with findings from experimental schistosome studies, as well as observed parasite-related morbidity, especially malnutrition, poor growth, and poor physical and cognitive performance in schistosome-infected children. Our findings further reiterate the need for control, early detection, and treatment of schistosome infections in preschool-aged children, and further mechanistic studies will inform development of interventions, for example nutraceuticals, in order to reduce disease consequences associated with schistosome infections. Thank you very much for your attention.
Brilliant. Thank you very much indeed, Derick. We've got a few questions that have popped up in the Q&A. So first of all, coming from Julio De Leo, asking about do you really think that it's a difference in the immune system with the difference between the two sectors, or do you think it's a consequence of exposure? So I'll let you answer that one first.
All right, thanks for the question. I would think it's the consequence of exposure and less of significant differences in the immune system. I think that the differences in the immune system will come to play post-exposure, where the immune system skewing starts to show marked differences between the two groups.
Yeah. And then Carlos has asked a question about the diagnostic methods and what you use for the definition of true infected children. Right.
So that one of the main limitations of the study is the fact that we used pathological methods for diagnosis, which we know has its own limitations in terms of sensitivity and all of that, especially in preschool-aged children. However, at the time we conducted the study, pathological methods still remain the gold standard, and so we feel that it is still plausible that we did use that. For true infections, certainly in terms of exposure and all of that, we agree that there will be more sensitive methods in determining that. However, we made sure this pathological diagnosis was in conjunction with guardian interviews, review of clinical history, administering of questionnaires to ensure that they had never been treated or been involved in any form of MDA. And so these together were what we used to, especially at the beginning, define people who had never been infected. And so following up using the pathological diagnosis was sufficient.
Okay, brilliant. There's also a question from Anna Kildemos here, who's asking about the metabolite profiles. And and are you, I mean, you might not be aware of the Balog paper et al. That she references, and wondering if that you could narrow down a set of predictors and if what you see as your profiles are similar to what's previously been published.
Yes, so especially in relation to what has previously been published, I think that our findings corroborate with that of especially experimental studies and a few human studies. At least I can recollect 2, but mostly in S-Mansonai. And the link with energy metabolism especially cuts across most of the studies that have been conducted with regards to metabolite profiles. Yeah.
Okay, brilliant. And if we've got time for a couple more, there's a couple of questions from the panelists. So for all the other people, we can't actually put them into the Q&A, so they're going into the chat. But fascinating work. And Nick is curious, do you think the upregulated metabolites could be a general marker of infection, immune activation, or specific to schistosome infections?
Tricky. I'm sure you know we can do some analysis and use that as markers, but for schisto personally, I believe these kinds of studies are more important in terms of understanding what the host parasite interactions are, to use them as biomarkers, especially when it comes to the economics, the feasibility, and all of that. You run into the challenge of it being too specialized to want to use that as some kind of marker. Although it's important, I personally feel, given the nature of the disease and the scope and the region and all of that, in terms of logistics, this is good in understanding where we're at, what is going on, and to inform us developing more specific interventions to reduce the morbidity that is associated with infection.
Okay, brilliant, thanks. And I guess that it's reverse post treatment with prazoquantel could lend strength in schistosome specific.
Yes. Yes,
there is another question from Fiona, but Fiona Allen, do you want to pop it so that everybody can see it, and then maybe Derick can reply in the chat so that everyone can see the answer, and that's quite kind. It might also be linked. But I think in the interest of time, we need to move on. But thank you very much again, Derick.
Thanks.
And, now I'm delighted to introduce our final speaker of the day, Jing-Bo Zhu, who is a research assistant at the CDC in China. And he's going to be talking about the potential impact of flooding on schistosomiasis in the Poyang Lake regions, based using multi-source remote sensing imaging. So thank you very much, and over to you, Jing-Bo.
Hello everyone, my name is Jing-Bo, and I'm working in National Institute of Parasitic Diseases, China CDC. And I'm very happy to share our recent research, potential impact of floods on schistosomiasis in Poyang Lake regions based on multi-source remote sensing images. Schistosomiasis is transmitted by snails as the only intermediate host. The occurrence and distribution of snails directly affect the prevalence and transmission of schistosomiasis. The distribution of snails is closely related to national conditions such as temperature, light, rainfall, and humidity. The Yangtze River has been hit by continuous heavy rainfall since June 2020, leading to a nationwide alert for water levels along the river and its tributaries, with many places exceeding the peak water level of the 1998 floods. The flood disaster caused the riverbank break and river water backflow, which increased the flooded area. Snails could spread to the surrounding areas by floods or floating objects, especially after 2 to 3 years, when the snail expanded and redistributed after adapting to the new environment. Okay, this slide shows the biological characters of snails. The type of distribution is lake type, and the influence of water distribution is winterland and summer water. The time of medication and field survey in spring and autumn. The suitable breeding temperature between 20 to 25. The elevation is about 20 to 35 meters, and the vegetation covers the sunshine at just humidity, and the water is necessity. Okay, the figure A shows the environment suitable for snail breeding, and the figure B shows the environment unsuitable for snail breeding. Okay, this figure shows the location of survey points, and this survey data will be used to improve the snail remote sensing model and verify the effectiveness of the model in the dynamic monitor of snail distribution. Okay, what parameters we can get from the remote sensing? It includes the NDVI and the wetness and the brightness and the modified normalized different waters. So in this research, we choose the Poyang Lake as our study area, and the Poyang Lake is the largest freshwater lake in China. It's located in the middle and lower reaches of the Yangtze River in the North Jiangxi province. And the multi-source remote sensing image, we chose the Landsat 8 satellite and Sentinel-1 images. The snail data distribution were obtained from a snail survey carried out in Jiangxi province in 2016. Okay. In this study, the distribution of snail habits was estimated using Landsat 8 satellite remote sensing image data, and areas where water body overlapped before and after flooding were identified using Sentinel-1. image data. So we use the double polarization threshold segmentation method to extract the water body, and the figure shows the changes in the area of water bodies before and after flooding in the Poyang Lake areas, and the blue regions indicate the appearance of water body on 15 May 2020 before the floods, and the red regions indicate the expansion in the area of the water bodies on 16 July during the flood period. So the left figure shows the distribution of snail habits in Poyang Lake areas. NDVI data were used to measure the vegetation cover in snail habits collected from Landsat 8 remote sensing images. And the right figure shows the prediction of snails' habits based on flood-affected setting. Snail spread was predicted to possibly cover an area of 759 square kilometers and to mean occur east of Yongxiu, south of Lushan, southwestern Poyang and Duchang, northwestern Xinjiang and Yugan. And then we get the risk map. The risk classification of spread of snails. The NDVI value of suitable snail habits were collected based on snail density data, and the risk was classified as higher density, medium density, and low density snail habits with reference to the NDVI values. So in this research, we get the conclusion: the snail breeding environment and its adjacent areas are submerged or scarred by floods, which may lead to the formation of new breeding environment for snails, further impacting the snail area and increasing the risk of schistosomiasis transmission. And it is feasible to identify the risk area of the schistosomiasis after floods based on water information extraction from multi-source remote sensing image data. And it is suggests that schistosomiasis control institutions should carry out snail surveillance and investigation on the distribution of infection source in the next 2 or 3 years. Okay, that's all. Thank you for your attention.
Thank you very much indeed. Do we have any questions for Jing-Bo? Any from the panelists as well? We don't think we got anything in the Q&A. Okay. Well, I mean, thank you. Sorry, I was thinking. I mean, what we can do in the interest of time is just wrap up, and if we do have any questions, we can also ask those tomorrow as well. So thank you very much again for that great talk and for all the wonderful work there in China. So thank you to everybody really for attending the first day of this meeting. It's been brilliant. We've heard from the research working groups, sorry, the working groups, not the research, that's my bias, the working groups and the work streams. We've heard about ultrasound work, about vaccines, about geospatial modeling, rotifers, and Japonicum population structure, and all sorts. I mean, future vector distributions, early infections, and their effect in preschool-aged children, as well as our last talk. So thank you. It's been really interesting. Tomorrow is going to be an equally interesting day. So we've got talked on the effect of sort of multiple intense praziquantel treatment and that effect on vaccine responses. We've got how to treat water using chlorination to kill off Cercaria. We're looking at diagnostics using glycan microarrays, but also looking at universal health coverage and looking at a more social science approach to how we can achieve equity. And then we've also got a really nice rapid fire early careers researchers session tomorrow, where we've got 7 different early career researchers giving 5-minute sort of blast talks on their research. So that's going to be really exciting as well. And then we've got a wonderful networking opportunity as well. So we will be sending a feedback form. There'll be a link sent after this meeting today. So please do give us feedback, which we can incorporate if we have time by tomorrow, but it's certainly for any future meetings. The link is the same for tomorrow, but then obviously when you get onto the link, you click on Wednesdays rather than Tuesdays. I'm just seeing some questions popping up in the Q&A whilst we go. So we have a little bit of time to play with. So I'm very happy to have some questions here. Hang on. Here you go. In China, you have such huge water bodies. What is the recommendation for molluscicides? So that's one question for you there. Unmute. I think she...
Okay. Can you hear me?
Yes, we can hear you. Thanks.
Oh, okay. Yes, there is many. There is a large water body, and the sorry.
Do you want me to copy the question? I mean, also Carlos. I mean, Xu, you can answer this, but the snails in China are slightly different to the snails in Africa in that they're only semi-aquatic. So a lot of the control for the snails in China for the Philippines and Japonicum transmitting is more vegetation removal. Is that right? Rather than mollusciding, because I believe with mollusciding they just crawl out of the water till it's all gone and then they go back in.
Yes.
But, yeah, I mean, with the huge water bodies, what are your recommendations for the snail control in those regions?
Because I don't do this mouse size, so I don't know how to use this.
It's fine. Maybe you are much more on the modeling of the remote sensing imaging, so rather than the actual snails themselves.
Yes.
Okay, that's fair enough. So if we have any more technical questions and more sort of statistical questions, then please do send them in for Xu. And have we got any more? There was one I think popped up for.
There's a question from Julio, if that's of interest. So he says, Dr. Xu, I think that your published paper is based on NDVI, while in this presentation you also used other indexes.
No.
Okay, no. It was NDVI.
Yeah, I just use NDVI for my index because the time is limitation, so I don't use the other index. Maybe in the next research, I will add the other index for temperature and other index to make our research.
Perfect, thank you. Hopefully, Julio, that has answered your question.
So he's just replied saying there was a slide with 4 indexes. And just for anyone else who's not sure, so NDVI is the Normalized Difference Vegetation Index, is that correct?
Yes, it's right.
Excellent. Yes, so I mean, certainly that's something that we can put you two in touch if you would like to discuss that further. And one question again, so from Carlos. Shan has performed modeling of climate change. Do you also have modeling of future possibilities of expansion of snails breeding sites in that area? So this is where we can bring two of our speakers together today already.
Yeah, Dr. Liu is in our is my colleague, and he has too many modeling for the climate changes. So maybe in the future, I will do ask him and to compare with to work with him and to use the climate change and to make our model to get better.
Yeah, no, that would be good, because I think with climate change, obviously you will also see increased flooding frequency. Is that right? And severity. So it might be, yeah, a really interesting.
Yeah.
And then maybe we can do some Brazil-China comparisons with future predictions of the snails and things. Brilliant. Do we have any other questions? I mean, we could ask Derick the question that I didn't have time to ask from Fiona Allen, which was about the effect of co-infection on your metabolite profiles.
Right. I mean, several other infections do, influence metabolite profile. I can't say for sure exactly what direction it would have went, but I'm certainly sure, at least more especially for energy, as we've seen, and it's influencing malnutrition and stunting and all of that is likely to be consistent, because in most of these endemic areas anyway, there are other, several other tropical diseases the children are co-infected with, which is even why we had to exclude that by screening and ensuring that. So yes, I'm sure it will change. I can't say exactly in what direction, but certainly for energy metabolism, which is key in most infections, I'm sure that will certainly still show. Yeah.
And Nick, you posted a comment just to the panelists as well. Do you want to mention that about your question sort of regarding, is it schistosome specific? I mean, I know we talked about it before, or infection specific, and if it changes afterwards, just following up on that discussion, because that might be something quite interesting that people might want to hear as well.
Yeah. I mean, my mind has sort of moved on, so let me think. Yeah, so my curiosity was, sort of went along with the co-infection idea. Do you know that those children that weren't infected with schistosomes also weren't infected with anything else? Like, do you have white blood cell counts from those children? And could you say, that, could you begin to get at a schistosome specific metabolite response that maybe would or would not be resolved after PZQ treatment and worm clearance?
To say we know for certain, especially at infection, no, but at least in the beginning, before recruitment, any child that was followed up, we made sure to screen for any other co-infections, review clinical data and clinical history, and exclude all of that. So yes, at 3 months, we don't have, like you said, full blood counts and WBCs and all of that. But at least in the study, the fact that all the alterations we saw were consistent with infection intensity, and the fact that these alterations reversed post-consequential treatment sort of go to confirm that the observations we made were due to that external stimulus, i.e. schistosome infection, and the kids were infected with. In terms of irreversibility, for what I know with the metabolic system, I'm sure that if we had followed up for a much longer period where, you know, morbidity and the adaptation of the metabolic system gets to a point where it, you know, it switches to just adapt to the current situation, maybe, you know, upon treatment, it may not be reversible. But the good thing is, if we're seeing this, then, hey, it's an indication that we need to screen quickly, act fast, and, you know, make sure that we don't get to the point of irreversibility.
Great, thank you very much. Well, I mean, I already wrapped up, but I can wrap up again. So if there are no more questions for today, then we are really looking forward to seeing you all again tomorrow. And thank you again to all of the speakers today, and for all of the organisers in particular, Anoop Guvras. Thank you for all of your help in putting this together. And yeah, as I said, we've got lots of exciting talks tomorrow, including the rapid fire early career researchers who are doing some really great stuff. So we really look forward to seeing you all again tomorrow. And please do fill in the feedbacks, because we'd love to just get each meeting better and better. And obviously with virtual ones, we're still all learning. So thank you all very much.